Welcome to our channel. We are so glad you found your way here. Let us help you fall asleep. Let's begin with the microbiome. The quiet world living inside you. Take a slow, deep breath in and release it. Let the weight of the day fade away as we embark on an unhurried, atmospheric journey through the wonders of the cosmos. Your mind is quiet. The universe is vast, and we are ready to explore. The microbiome encompasses the diverse community of microorganisms inhabiting various environments, including the human body. These microscopic entities include bacteria, viruses, fungi, and protozoa, all coexisting in a delicate balance within the human gastrointestinal tract. For example, trillions of these microorganisms play crucial roles in digestion, metabolism, and immune response. This region functions as a complex ecosystem where each organism contributes to the overall health and well-being of the host. The composition of the microbiome can vary significantly among individuals, influenced by factors such as diet, environment, and genetics. This diversity is essential as it fosters resilience against diseases and helps maintain homeostasis. Within this intricate network, certain bacteria break down complex carbohydrates, while others synthesize essential vitamins, demonstrating a level of cooperation that is vital for survival. Interactions between these microorganisms can lead to beneficial or detrimental outcomes. Disruptions in this ecosystem, often referred to as dysbiosis, can give rise to various health issues, including inflammatory diseases, allergies, and obesity. Understanding the microbiome's intricate workings sheds light on its profound impact on human health, suggesting that maintaining a balanced microbial population is fundamental. Research continues to explore the many nuances of this hidden world, revealing the essential interdependencies that define both the microbiome and its host. The exploration of microorganisms began in the 17th century when Antony van Leeuwenhoek first employed a microscope to uncover the unseen. His meticulous observations revealed a universe of tiny entities, which he referred to as animalcules. This discovery marked a pivotal shift in understanding life, as these microscopic organisms challenged the prevailing notions of biology during that era. By the 19th century, advancements in technology allowed scientists like Louis Pasteur and Robert Koch to delve deeper into microbial life. Pasteur's experiments debunked the theory of spontaneous generation, establishing that microorganisms could spoil food and cause diseases. This insight laid the groundwork for germ theory, fundamentally altering the landscape of medicine and hygiene. Koch's postulates further refined the understanding of pathogenic bacteria, providing a systematic approach to link specific microorganisms to particular diseases. These developments ignited a revolution in public health, leading to sanitation reforms and the development of vaccines. The early 20th century saw a burgeoning interest in the relationship between microorganisms and their environments. Research began to reveal that not only pathogens existed among these entities, but also beneficial organisms that played crucial roles in digestion, immunity, and overall health. This nuanced understanding continues to evolve as modern science probes deeper into the complexities of this intricate ecosystem within. The Human Microbiome Project stands as a landmark initiative aimed at mapping the diverse microbial communities residing within the human body. It endeavors to catalog the myriad species of bacteria, viruses, fungi, and other microorganisms that thrive in various environments, from the gut to the skin. A fundamental objective centers on establishing a comprehensive reference database, which will serve as a baseline for understanding how these microbial populations contribute to health and disease. Initial findings revealed that the human microbiome is far more complex than previously understood, comprising trillions of microorganisms that outnumber human cells. These microbial residents participate in essential processes such as digestion, vitamin synthesis, and immune regulation. Specific patterns of microbial composition have been correlated with a range of health conditions, including obesity, diabetes, and autoimmune disorders. The project has also illuminated the dynamic nature of these communities, influenced by factors such as diet, environment, and lifestyle. This variability underscores the importance of personalized approaches to health and medicine, as individual microbiomes can differ significantly from one person to another. Through advanced genomic sequencing techniques, researchers continue to uncover the roles of specific microbes, revealing their contributions to metabolic functions and overall well-being. Insights from the Human Microbiome Project promise to transform therapeutic strategies and enhance our understanding of the intricate relationship between humans and their microbial inhabitants. Bacteria, archaea, fungi, and viruses constitute the primary categories of microbiota residing within the human body. Each group plays a distinct role, contributing to the overall functionality of the microbiome. Bacteria, the most abundant representatives, engage in various processes, including digestion and immune modulation. They thrive in diverse environments, from the gut to the skin, and their interactions can influence health outcomes significantly. Archaea, less commonly known, inhabit extreme environments, yet they also find a niche within the human gut. Their metabolic processes can assist in breaking down complex carbohydrates, enabling energy extraction that may go unnoticed in typical dietary assessments. This capability highlights their importance in maintaining a balanced intestinal ecosystem. Fungi, often overshadowed by bacteria, possess their own unique functions. Yeasts, a common type of fungi, participate in fermentation processes and can impact overall gut health. While usually benign, an imbalance in these microorganisms can lead to dysbiosis, potentially resulting in various health issues. Viruses, particularly bacteriophages, engage in interactions with bacteria, influencing their population dynamics and genetic exchange. These entities may seem insignificant, yet their presence contributes to the complex interplay among microbiota, underscoring the delicate balance that defines human health. This microcosm reflects an intricate web of life operating quietly and efficiently within. Digestion serves as a vital process, facilitating the breakdown of food into absorbable nutrients. The gut microbiome plays an essential role in this intricate system, contributing to the fermentation of complex carbohydrates and the synthesis of key vitamins. This community of microorganisms thrives in an environment rich in nutrients, where they interact with dietary components and the host cells. In this region, bacteria such as Bacteroides and Lactobacillus engage in the breakdown of fibrous substances, converting them into short-chain fatty acids. These metabolites not only provide energy for intestinal cells, but also influence systemic inflammation and immune responses. Nutrient absorption is enhanced by the presence of these microorganisms, which help maintain the integrity of the intestinal barrier, ensuring that essential nutrients pass efficiently into the bloodstream. Microbial diversity within the gut correlates with a range of health outcomes. A balanced microbiome supports optimal digestion and nutrient uptake, while an imbalance may lead to gastrointestinal issues and malabsorption. The interactions between these microorganisms and dietary inputs extend beyond mere digestion, impacting metabolic pathways and even mood regulation. The gut microbiome exemplifies a complex interplay between the host and its resident organisms, reflecting a delicate balance crucial for overall health. This hidden ecosystem continues to reveal its significance, emphasizing a deeper understanding of the connections between diet, microbiota, and wellness. The skin microbiome serves as a protective barrier, playing a crucial role in immune function. Composed of diverse microorganisms, it forms a complex community on the skin's surface, working in concert to fend off pathogenic invaders. This region acts not only as a physical shield, but also engages in biochemical interactions that maintain skin health. Microorganisms residing here contribute to the production of essential antimicrobial peptides. These compounds help neutralize harmful bacteria, fungi, and viruses, reinforcing the skin's defense mechanisms. Additionally, they modulate inflammatory responses, ensuring that the immune system reacts appropriately to threats without overreacting. Such balance is vital for preventing skin disorders, which can arise from dysbiosis, a disruption in microbial equilibrium. Restoration of the skin microbiome can be influenced by various factors, including diet, environmental conditions, and hygiene practices. Probiotics and prebiotics are increasingly recognized for their potential to support this delicate ecosystem. When applied topically or ingested, they may enhance microbial diversity and strengthen the immune response. Recent research highlights the interconnectedness of the skin microbiome with overall health. Its influence extends beyond mere protection. It plays a role in conditions like eczema, acne, and even systemic diseases. Understanding its intricacies may lead to innovative approaches in skincare and disease prevention, showcasing the profound impact of this hidden world. The oral microbiome plays a crucial role in maintaining dental health and has broader implications for systemic diseases. This complex ecosystem, teeming with diverse microorganisms, resides in the mouth, where its balance is essential for preventing conditions such as gingivitis and periodontitis. The presence of beneficial bacteria helps to inhibit the growth of harmful pathogens, creating a protective barrier against decay and infection. When the delicate equilibrium of this environment is disrupted, a cascade of effects may ensue. Dysbiosis, the imbalance of microbial communities, can lead to inflammation and an increased susceptibility to dental diseases. Research indicates that oral bacteria can translocate into the bloodstream, which may contribute to systemic issues such as cardiovascular disease and diabetes. This connection underscores the significance of maintaining oral health, as it is intertwined with overall well-being. The oral microbiome's influence extends to digestion and the immune system. Metabolites produced by these microorganisms can affect nutrient absorption and modulate inflammatory responses throughout the body. Exploring the relationship between oral health and systemic conditions reveals the potential for targeted interventions that leverage the microbiome. Dietary choices, oral hygiene practices, and probiotic applications may play pivotal roles in fostering a balanced microbial community. The vaginal microbiome significantly influences reproductive health, serving as a crucial line of defense against pathogens. This region is predominantly inhabited by lactobacillus species, which help maintain an acidic environment, deterring harmful bacteria and yeast. When the balance of these microorganisms is disrupted, conditions such as bacterial vaginosis or yeast infections may arise, leading to discomfort and potential complications. In a healthy state, the presence of lactobacillus aids in the production of lactic acid, creating a protective barrier. This equilibrium is essential not only for general well-being, but also for reproductive processes. A well-maintained microbiome can contribute to optimal fertility, facilitating the establishment of a healthy pregnancy. Conversely, an imbalanced microbiome may affect sperm motility and viability, increasing the risk of infertility. Interactions between the vaginal microbiome and the immune system are intricate. Pathogens can provoke inflammatory responses, influencing overall reproductive health. Infections may also lead to complications during pregnancy, such as preterm birth or low birth weight. Research continues to explore the application of probiotics in restoring balance within this microbial community, with the hope of enhancing reproductive outcomes. Further understanding of this intimate ecosystem may offer new avenues for treatment and prevention, reinforcing the importance of microbial harmony in reproductive health. Microbiome diversity plays a crucial role in maintaining health, as a rich array of microbial species contributes to stability and resilience within this ecosystem. An abundance of different species ensures that the community can respond effectively to environmental changes, such as fluctuations in diet or exposure to pathogens. Each species, from bacteria to fungi, possesses unique functions of base, allowing for a complex interplay that enhances metabolic processes, immune responses, and nutrient absorption. In this region of the microbiome, specific microbial populations work synergistically, fostering an environment that discourages pathogen colonization. Species richness is integral to this process, as a more varied microbiome can outcompete harmful microorganisms for resources and space, thereby reducing the risk of infections. Research indicates that diminished diversity is often associated with various health issues, including obesity, diabetes, and gastrointestinal disorders. These findings suggest that maintaining a diverse microbiome may be a key factor in preventing disease. Restoration of microbial diversity can often be achieved through dietary changes, including the introduction of prebiotics and probiotics. This adjustment not only supports the growth of beneficial species, but may also reduce the dominance of harmful ones. In this continuous cycle of interaction, the microbiome exemplifies the delicate balance required for health, emphasizing the significance of species richness in sustaining well-being. Genetics plays a pivotal role in shaping the microbiome, influencing the types and abundances of microorganisms that inhabit the body. Specific genes can affect immune responses, determining how the body interacts with various microbial communities. Variations in the host genome can lead to distinct microbial profiles, contributing to individual differences in health and disease susceptibility. This intrinsic factor, while significant, is complemented by external elements that further dictate composition. Though environment acts as a critical variable, encompassing dietary habits, lifestyle choices, and geographic location, nutritional intake directly impacts microbial diversity, with fiber-rich diets promoting a flourishing ecosystem of beneficial bacteria. Conversely, highly processed foods may diminish microbial richness, leading to potential health consequences. Interactions with the environment extend to exposure to pollutants and antimicrobial agents, which can disrupt the delicate equilibrium within the microbiome. Additionally, early life experiences, including mode of birth and breastfeeding, set foundational microbiome characteristics. These early microbial colonizers establish a baseline that can influence health trajectories throughout life. The interplay between genetics and environmental factors creates a dynamic landscape within the microbiome, continually evolving in response to various stimuli. Understanding these influences provides insight into the complex relationship between human health and the microorganisms that reside within. Food choices have a profound impact on microbial communities residing in the gastrointestinal tract. Different diets promote the growth of distinct microbial populations, which in turn can influence metabolic processes, immune responses, and overall health. A high-fiber diet, for instance, supports the proliferation of beneficial bacteria, which thrive on complex carbohydrates. These microorganisms produce short-chain fatty acids during fermentation, contributing to gut health and systemic inflammation reduction. Conversely, diets rich in processed foods and sugars can lead to a decline in microbial diversity. This region of the gut may become dominated by harmful bacteria, potentially increasing the risk of metabolic disorders and inflammatory diseases. The delicate balance within these communities is sensitive to dietary changes, suggesting that even small adjustments in nutrition can yield significant effects on health. Fermented foods, such as yogurt and kefir, introduce live beneficial microorganisms, enhancing the richness of the microbiome. Regular consumption of these foods can fortify the diversity and abundance of beneficial species, strengthening the gut barrier and immune function. Seasonal variations in food availability further highlight the adaptability of these microbial communities. Different regional food sources can lead to unique microbial signatures, illustrating the dynamic interplay between diet and the living ecosystem within the human body. This intricate relationship underscores the importance of mindful dietary choices. The impact of antibiotics on microbial diversity is profound and multifaceted. These substances, designed to combat harmful bacteria, inadvertently alter the delicate balance of the microbiome. When introduced into the body, they do not discriminate between pathogenic and beneficial organisms. This leads to significant shifts in microbial populations, often resulting in decreased diversity. Such reductions can compromise the microbiome's ability to perform essential functions, including digestion and immune response. In this altered state, certain species may become dominant while others diminish, creating a less resilient ecosystem. Studies indicate that a single course of antibiotics can lead to a long-lasting decrease in microbial richness. This disruption is not merely temporary. Some changes may persist for months or even years following treatment, emphasizing the importance of cautious antibiotic use. The repercussions extend beyond individual health. Altered microbial communities can influence overall well-being, contributing to conditions such as obesity, allergies, and autoimmune diseases. The interconnectedness of these organisms suggests that maintaining microbial diversity is crucial for health. In light of these findings, researchers advocate for more judicious use of antibiotics, urging a balanced approach to treatment. Understanding the impact of these agents on microbial ecosystems may pave the way for strategies to preserve and restore microbial health in the future. The role of probiotics and prebiotics within microbial ecosystems represents a crucial aspect of maintaining microbial health. Probiotics are defined as live microorganisms that, when administered in adequate amounts, confer a health benefit to the host. These beneficial bacteria, such as Lactobacillus and Bifidobacterium, can enhance gut health, support immune function, and even influence mental well-being through the gut-brain axis. Prebiotics, on the other hand, serve as food for these microorganisms. They are typically non-digestible fibers found in various plants, such as chicory root, garlic, and bananas. By promoting the growth and activity of beneficial bacteria, prebiotics play an essential role in shaping the composition of the microbiome. This interaction between probiotics and prebiotics highlights the delicate balance required to sustain a healthy microbial community. The distinction between the two is fundamental yet interconnected. Probiotics introduce live organisms that can positively affect health, while prebiotics nourish and enhance the existing microbial population. Together, they contribute to the resilience and diversity of the microbiome. Research continues to explore the potential of these agents in various health conditions, from gastrointestinal disorders to metabolic diseases, underscoring the significance of nurturing this vibrant and visible world that resides within. Maintaining equilibrium in these populations remains a vital aspect of overall health. Fermented foods serve as a significant means of fostering beneficial microbiota in the gut. These foods undergo a process of fermentation, during which microorganisms such as bacteria and yeast transform sugars and starches into acids, gases, and alcohol. This transformation not only preserves the food, but also enhances its nutritional profile and bioavailability of various compounds. Kefir, sauerkraut, kimchi, and yogurt are well-studied examples, rich in probiotics that actively support gut health. The introduction of live cultures from these foods encourages the growth of beneficial bacteria, creating a diverse microbial community, essential for the maintenance of gut integrity. Consuming these foods regularly can assist in restoring balance, particularly after disruptions caused by antibiotics or dietary changes. The metabolic products of these microorganisms can influence various bodily functions, including digestion, immune response, and even mood regulation. These fermented offerings contribute to the production of short-chain fatty acids, which provide energy to colon cells, while also playing a role in reducing inflammation. Incorporating a variety of these foods into the diet may enhance microbial diversity. This diversity is critical, as it is linked to improved resilience against pathogens and overall health. Insights into this relationship continue to inform dietary guidelines, showcasing the importance of nurturing the microbiome through natural sources. The gut-brain axis represents a complex communication network that links the gastrointestinal system and the central nervous system. This connection reveals how the microbiome influences mental health, with emerging studies suggesting that gut bacteria can produce neurotransmitters, such as serotonin, which significantly affect mood and behavior. It is within this intricate dialogue that the microbiome's role becomes increasingly critical, as alterations in microbial composition can contribute to mental health disorders. Research indicates that certain microbial strains may enhance cognitive function and emotional well-being, while others may be associated with anxiety and depression. This region of the human body serves not only as a digestive powerhouse, but also as a conduit for emotional regulation. Individuals with an imbalance in their microbiome may experience disruptions in this delicate balance, leading to various psychological conditions. Interventions aimed at restoring microbial health, such as probiotics and dietary modifications, show promise in alleviating symptoms of mental health disorders. The influence of gut microbiota extends beyond simple digestion. It encompasses a profound impact on overall psychological resilience. Understanding these connections deepens the comprehension of mental health, emphasizing the need for a holistic approach to treatment that considers both the gut and the brain. This ongoing research continues to unveil the depths of human health, driven by the unseen inhabitants within. Interactions between the microbiome and the immune system reveal a delicate balance essential for maintaining health. These microorganisms reside primarily in the intestines, where they play a pivotal role in shaping immune responses. The presence of beneficial bacteria fosters a state of homeostasis, allowing the immune system to differentiate between harmful pathogens and harmless entities. This intricate relationship is governed by a complex signaling network, microbial metabolites, produced during the digestion of dietary fibers, communicate directly with immune cells. These interactions promote the development of tolerance, ensuring that the immune system does not overreact to benign substances, which could lead to autoimmune disorders. Diversity within the microbiome enhances the robustness of this system. A varied population of microbes contributes to a broader range of metabolites and immune modulation, providing resilience against infections and diseases. Conversely, dysbiosis, an imbalance in microbial populations, can compromise immune function, leading to inflammation and increased susceptibility to illnesses. Research indicates that environmental factors, such as diet, stress, and antibiotic use, can significantly influence microbiome composition. Adjustments in these factors can provoke rapid shifts in microbial communities, affecting immune system stability. Understanding these interactions is vital for developing strategies that promote health through microbiome management and restoration. Dysbiosis refers to an imbalance within the microbiome where beneficial microorganisms are outnumbered by harmful or pathogenic species. This disruption can result from various factors, including poor diet, stress, antibiotic use, and environmental influences. In such cases, the harmonious relationship between host and microbes is compromised, leading to a cascade of physiological consequences. The manifestations of dysbiosis can be wide-ranging. It often correlates with gastrointestinal disorders, such as irritable bowel syndrome and inflammatory bowel diseases. In this region of the body, the delicate equilibrium is crucial for digestion, nutrient absorption, and immune function. When disrupted, the resulting inflammation can impair these processes, leading to discomfort and malabsorption issues. Beyond the gut, the ramifications extend to other systems. Emerging research indicates links between dysbiosis and conditions such as obesity, diabetes, and even mental health disorders. The gut-brain axis, a pathway of communication between the gastrointestinal tract and the central nervous system, highlights how microbial imbalances can influence mood and cognitive functions. Restoring balance within this microbial community is essential. Strategies such as dietary modifications, probiotics, and prebiotics aim to replenish beneficial species and suppress harmful ones. A comprehensive understanding of dysbiosis and its consequences is necessary for fostering overall health and well-being. Chronic stress influences the delicate balance of the microbiome, shifting the composition of these communities that reside within it. Elevated cortisol levels, often a byproduct of prolonged stress, can lead to a decrease in beneficial bacteria. This shift allows for the overgrowth of opportunistic pathogens, creating an environment conducive to dysbiosis. The gut, often referred to as the second brain, communicates bidirectionally with the central nervous system. This connection means that a microbiome under duress can further exacerbate feelings of anxiety and depression, forming a feedback loop that complicates overall mental health. In response to stress, these microbial changes may also affect gut permeability, leading to a condition commonly referred to as leaky gut. This phenomenon allows toxins and undigested food particles to enter the bloodstream, potentially triggering inflammatory responses. In this region of the body, The interplay between stress and microbial health highlights the importance of maintaining emotional well-being for physical health. Various studies indicate that practices aimed at stress reduction, such as mindfulness and regular physical activity, may help restore microbial balance. These interventions appear to play a crucial role in mitigating the adverse effects of stress on gut microbiota. Observations suggest that fostering a healthy microbiome can contribute to enhanced resilience against stress-related disorders, promoting a state of equilibrium within the host. The early stages of life are marked by a crucial process of colonization and development, where unique microbial communities begin to establish themselves. This initial colonization occurs primarily during birth, as infants encounter diverse microbial environments from their mothers and surroundings. Factors such as mode of delivery play a significant role in shaping the composition of early microbial inhabitants. Vaginal births typically expose infants to maternal flora, while cesarean sections may result in a different microbial profile. During the first few months, exposure to various environmental sources, including family members and pets, further enriches this developing microbiome. Breastfeeding provides not only essential nutrients but also prebiotics that support the growth of beneficial bacteria. The interplay between the host and these microbes begins to form a delicate balance, aiding in digestion and influencing immune responses. As the child grows, dietary changes introduce new microbial species, leading to an evolving ecosystem. The diversity of this microbial community is crucial for long-term health, as it establishes a foundation for metabolic processes and immune system training. Disruptions during these formative years, such as antibiotic use, can hinder the natural progression of microbiome development. Awareness of these early influences underscores the importance of nurturing this internal environment, which remains pivotal throughout life. The maternal microbiome plays a crucial role in shaping fetal development. This internal ecosystem establishes foundational health parameters, influencing immune system formation and metabolic pathways. Microorganisms in the maternal body communicate with the developing fetus, facilitating essential nutrient absorption and even impacting gene expression. During pregnancy, maternal microbiota undergo dynamic changes, reflecting adaptations to support both the mother and growing child. These shifts create a unique microbial environment, which can either promote health or predispose to complications. The presence of specific bacteria can enhance the transfer of vital compounds, fostering optimal growth conditions for the fetus. Research highlights how the maternal microbiome may contribute to the development of the fetal gut microbiome. As the fetus transitions from a sterile environment to an external world teeming with microbial life, the maternal influence becomes evident. This introduction paves the way for establishing a diverse and resilient microbiome that can withstand environmental perturbations. Imbalances in the maternal microbiome have been associated with various pregnancy complications, such as preterm birth and gestational diabetes. Understanding these connections emphasizes the potential for targeted interventions to improve maternal and fetal health outcomes. This intricate interplay between maternal and fetal microbiomes continues to reveal complexities that challenge previous assumptions about human development and health. Microbial inheritance plays a significant role in shaping the microbial communities that inhabit an individual. These communities are influenced by family dynamics, genetic relationships, and environmental exposures. The process begins at birth, as the initial colonization of the microbiome is heavily dictated by the method of delivery. Vaginal births expose infants to a diverse array of maternal microbes, establishing a foundation that closely resembles the mother's microbiome. In contrast, those born via cesarean section often encounter a different microbial landscape. The absence of maternal bacteria at the time of delivery may lead to distinct microbial compositions, influencing health trajectories from the outset. Family interactions further shape microbial inheritance. Close contact among family members facilitates the transfer of microbes, establishing shared microbial environments. These connections can lead to similarities in microbial profiles among siblings, as they share not just genetics but also living spaces, dietary habits, and lifestyle. The environment significantly contributes to the microbiome's development, with shared living conditions resulting in greater microbial overlap. As individuals grow, the microbiome continues to evolve, reflecting changes in diet, health, and external exposures. The delicate balance between inherited and acquired microbes underscores the dynamic nature of this ecosystem. Understanding these relationships enhances our knowledge of how microbial communities influence health across generations, revealing a complex lineage of microbial inheritance that persists throughout life. The microbial landscape within the human body undergoes significant transformations as aging progresses. In youth, a diverse and abundant array of microbes flourishes, creating a robust ecosystem that supports various physiological functions. This diversity is crucial for metabolic processes, immune system development, and overall health. As time passes, these microbial communities begin to shift, often exhibiting a decrease in richness and variability. The decline in microbial diversity can be attributed to several factors, including dietary changes, environmental exposures, and alterations in lifestyle. These influences contribute to a less stable microbiome, which may become increasingly dominated by specific microbial taxa. Such shifts have been associated with age-related health issues, including inflammation, metabolic disorders, and cognitive decline. Research indicates that certain beneficial microbes tend to diminish with age, leading to reduced production of essential metabolites that support bodily functions. Conversely, pathogenic species may proliferate, introducing potential health risks. This dynamic underlines the importance of maintaining microbial health throughout the lifespan. Interventions such as dietary modifications, probiotics, and prebiotics may help restore microbial balance, promoting a more diverse microbiome. Understanding these shifts in microbial composition provides insight into potential strategies for enhancing health and longevity, illustrating the intricate connection between microbial diversity and aging. Microbial composition influences metabolic processes, affecting energy extraction and storage within the human body. A diverse microbiome plays a crucial role in regulating the efficiency of nutrient absorption, which directly impacts weight management and the risk of metabolic disorders. Specific bacteria within this ecosystem are adept at breaking down complex carbohydrates and fibers, producing short-chain fatty acids that contribute to energy regulation and insulin sensitivity. This region of microbial activity becomes particularly significant in the context of obesity and diabetes. Imbalances in microbial populations can lead to excessive energy harvest, resulting in increased fat storage and the subsequent development of insulin resistance. Alterations in the gut microbiome have been consistently associated with obesity, suggesting a direct correlation between microbial health and metabolic function. Studies indicate that individuals with obesity often display reduced microbial diversity, a condition that may exacerbate metabolic dysfunction. Conversely, restoring a balanced microbial community through dietary interventions or probiotics has shown potential in managing weight and improving insulin sensitivity. Research continues to explore the mechanisms through which these microorganisms influence systemic metabolism, emphasizing the need for a deeper understanding of gut health. This knowledge may pave the way for novel therapeutic approaches targeting the microbiome to combat obesity and diabetes, reshaping the landscape of metabolic health. Emerging research suggests a significant relationship between the microbiome and cardiovascular health, This connection is increasingly recognized as the gut microbiota influences various processes within the cardiovascular system. Specific metabolites produced by these microorganisms, such as short-chain fatty acids, have demonstrated potential roles in regulating blood pressure and improving endothelial function. Elevated levels of certain microbial byproducts may contribute to the development of atherosclerosis, a condition characterized by the buildup of plaques in the arteries. In this context, the composition of the microbiome may serve as a pivotal factor in determining cardiovascular risk. Diverse microbial populations are associated with beneficial outcomes, while dysbiosis, or microbial imbalance, correlates with increased inflammation and higher cholesterol levels. Research indicates that dietary choices can modulate this microbial landscape, underscoring the importance of nutrition in maintaining cardiovascular health. Ongoing investigations are exploring the feasibility of microbiome-targeted interventions, which may involve prebiotics, probiotics, or dietary adjustments aimed at restoring microbial balance. These strategies hold promise for mitigating cardiovascular disease, potentially offering new avenues for prevention and treatment. Understanding the intricate interplay between gut health and cardiovascular function could redefine approaches to heart disease, highlighting the microbiome's role as a critical player in overall health and well-being. The connection between the microbiome and autoimmune diseases unveils a complex relationship that influences the immune system's behavior. Within this intricate ecosystem, diverse bacterial populations contribute to immune regulation. Disruptions in this balance can lead to the immune system mistakenly attacking the body's own tissues, a hallmark of autoimmune disorders. Certain strains of bacteria have been linked to the modulation of inflammatory responses, suggesting that they play a protective role. Meanwhile, an overabundance of pathogenic bacteria may incite inflammatory pathways, further exacerbating conditions such as rheumatoid arthritis and multiple sclerosis. Studies indicate that individuals with these diseases often display distinct microbiome profiles, highlighting the potential for targeted interventions. And the SUMF, restoring microbial diversity through dietary changes or probiotics, emerges as a promising avenue for research. The reintroduction of beneficial bacteria could mitigate the hyperactive immune responses characteristic of autoimmune diseases. Probiotic therapies aim to recalibrate the immune system, fostering a more balanced interaction that protects against self-reactivity. This region of study fosters hope for new therapeutic strategies, shifting from symptom management to addressing underlying microbial imbalances. Understanding these interactions emphasizes the importance of the microbiome, not merely as a bystander but as a key participant in the immune landscape, potentially reshaping the future of autoimmune disease management. Emerging research suggests intriguing connections between the microbiome and cancer. This intricate community of microorganisms inhabiting various bodily niches may influence tumor development and progression by modulating immune responses. They could either suppress or promote carcinogenesis, demonstrating a dual role that complicates their study. Certain bacterial species have been associated with increased cancer risk, while others appear to exert protective effects. For instance, the presence of specific gut bacteria may affect the metabolism of dietary compounds, leading to the production of metabolites that influence cellular behavior. These metabolites can either foster a favorable environment for cancer cells or inhibit their growth. Mechanisms underlying these associations are becoming clearer. Changes in the microbiome can alter the gut barrier function, allowing for translocation of harmful substances that may provoke inflammation or direct interactions with host cells. Inflammation, a well-known precursor to many cancers, can be influenced by microbial composition, thus linking these inhabitants to disease progression. Clinical studies are beginning to explore how manipulating the microbiome through diet, probiotics, or antibiotics may enhance cancer therapies. This research could redefine treatment approaches by integrating microbial health into cancer management strategies. As understanding deepens, new avenues for prevention and therapy may emerge, harnessing the potential of this internal ecosystem. The internal ecosystem plays a significant role in the modulation of immune responses. This region houses trillions of microorganisms, each contributing to the delicate balance of health. Changes in this balance can influence the development of various immune-related conditions, including allergies and asthma. Certain strains of bacteria may help to educate the immune system, promoting tolerance to harmless substances. When this education is disrupted, it can lead to heightened sensitivity, resulting in allergic reactions. Asthma, a chronic respiratory condition characterized by inflammation and constriction of the airways, has also been linked to the composition of these microbial communities. Research indicates that individuals with asthma often exhibit distinct microbiome profiles compared to those without the condition. Factors such as antibiotic use, diet, and environmental exposures can significantly alter these profiles, potentially exacerbating respiratory issues. In early childhood, the establishment of a healthy microbiome may offer protection against asthma development. Interventions aimed at restoring microbial diversity could hold promise for reducing incidence rates. Investigations continue into specific bacteria that might serve as beneficial agents in managing or preventing these conditions. By understanding the interactions between the microbiome and the immune system, new strategies may be developed to address the rising prevalence of allergies and asthma in modern populations. The connection between the microbiome and skin conditions such as eczema and psoriasis reveals a complex interplay of factors influencing health. In this region of the body, diverse communities of microorganisms inhabit the skin's surface, contributing to its barrier functions and immune responses. Research indicates that an imbalance in these microbial populations can exacerbate inflammatory skin disorders. Eczema, often characterized by red, itchy patches, may arise when beneficial bacteria decline, allowing pathogenic species to flourish. This disruption leads to inflammation and compromised skin integrity. In contrast, psoriasis presents as thick, scaly plaques resulting from accelerated skin cell turnover and immune dysregulation. The microbiome's role in modulating these processes is increasingly under investigation. Studies have shown that individuals with psoriasis often exhibit distinct microbial profiles compared to healthy skin. Therapeutic approaches targeting the microbiome could restore balance, helping to alleviate symptoms. Probiotic treatments and topical applications designed to enrich beneficial microbes are among the strategies being explored. The skin's ecosystem serves not only as a barrier against external threats, but also as a reflection of internal health. By delving into these microbial communities, researchers aim to unlock new avenues for treatment, addressing the underlying causes of these challenging skin conditions rather than merely managing their symptoms. This exploration continues to unveil the intricate relationships that define human health. Geography and culture play pivotal roles in shaping microbiome diversity. Each region, with its unique environment, contributes to a distinct microbial landscape. Variations in climate, soil composition, and the availability of local flora create a niche for specific microorganisms. These distinctions are further amplified by cultural practices surrounding food, hygiene, and medicine. For instance, diets rich in fermented foods promote the proliferation of beneficial bacteria, while urban living may expose individuals to different microbial exposures than rural environments. In diverse geographical settings, communities maintain unique traditions that influence their microbiomes. For example, the consumption of traditional foods rich in prebiotics can enhance microbial diversity, fostering a resilient gut environment. This resilience becomes increasingly significant in a world facing challenges such as antibiotic resistance and lifestyle diseases. Cultures that emphasize communal eating may facilitate the transfer of microorganisms among individuals, further contributing to diversity. This sharing creates a dynamic ecosystem within the human body, embodying the intricate web of interactions that define health and wellness. Understanding these geographical and cultural influences provides insights into the broader implications for public health. By appreciating the significance of local practices and environments, it becomes possible to tailor interventions that support microbiome health while respecting cultural identities. This nuanced approach fosters a deeper awareness of the interconnectedness between humanity and its microbial companions. Urban environments present a unique backdrop for microbial communities, characterized by heightened human activity and altered landscapes. These regions tend to exhibit reduced biodiversity compared to their rural counterparts. Factors such as pollution, increased temperature fluctuations, and limited vegetation play significant roles in shaping these microbial ecosystems. The density of human presence influences the composition of bacteria, fungi, and viruses, often leading to an abundance of opportunistic pathogens that thrive in disturbed environments. Contrastingly, rural settings frequently offer a more stable and diverse microbiome. Natural habitats, with their varied flora and fauna, provide a wealth of niches for microorganisms. Soil richness, vegetation cover, and the absence of industrial pollutants contribute to a more balanced microbial community. The interaction between animals and plants within these ecosystems fosters resilience, allowing for a dynamic exchange of microorganisms. Seasonal variations also play a critical role in shaping these communities. In rural areas, the changes in climate can influence microbial diversity, resulting in unique seasonal populations. In urban landscapes, the constant human influence often disrupts these natural cycles, leading to a more homogeneous microbial population. Understanding the disparities between urban and rural microbiomes sheds light on the profound impact of environment on microbial diversity. Recognizing these differences can inform public health strategies and enhance our grasp of the essential roles microorganisms play within various ecosystems. The interplay between the microbiome and inflammation reveals a complex relationship that influences health. In this region of the human body, microorganisms contribute to immune regulation and the maintenance of homeostasis. Specific bacterial populations can either suppress or exacerbate inflammatory responses, depending on their composition and abundance. Certain microbes produce metabolites that promote the production of anti-inflammatory cytokines, creating a protective environment against excessive inflammation. These metabolites, derived from dietary fibers, facilitate communication between gut bacteria and immune cells, fostering a balanced response. Conversely, dysbiosis, an imbalance within the microbial community, often leads to an increase in pro-inflammatory factors. This shift can trigger chronic conditions, including inflammatory bowel disease and other autoimmune disorders. Research indicates that the presence of specific microbial species correlates with the severity of inflammatory responses. An increase in pathogenic bacteria may result in heightened inflammation, while a diverse and stable microbiome appears to mitigate such risks. This relationship emphasizes the importance of microbial diversity for maintaining a healthy immune system. Vertiometra strategies targeting the microbiome, including probiotics and dietary adjustments, are gaining traction as potential interventions for managing inflammation. Understanding how these microorganisms interact with the immune system could pave the way for novel treatments aimed at restoring balance and reducing the burden of inflammation-related diseases. Interactions between the microbiome and pathogens reveal a complex ecosystem where beneficial microorganisms can either inhibit or facilitate the establishment of infectious diseases. This region of the human body serves not only as a habitat for trillions of bacterial cells, but also as a frontline defense against invading pathogens. The presence of these microorganisms often modifies the local environment, producing substances that can neutralize harmful bacteria and viruses. And in certain situations, however, disruptions in microbial communities can create opportunities for pathogenic organisms to thrive. Such imbalances may lead to conditions where the immune response is compromised, allowing infections to take hold more easily. These disruptions can arise from various factors, including antibiotic use, which indiscriminately alters microbial populations, or changes in diet that affect microbial diversity. Research continues to uncover the mechanisms through which the microbiome influences susceptibility to infectious diseases. For instance, specific strains of bacteria have been shown to produce metabolites that enhance the immune system's ability to respond to pathogens. In this context, the microbiome does not merely exist as a passive inhabitant, but actively participates in a dynamic interplay that shapes health outcomes. The implications of these findings extend beyond understanding disease mechanisms. They open new avenues for therapeutic interventions that leverage the microbiome to bolster defenses against infections. Microbiome sampling techniques offer a window into this intricate world, allowing researchers to gather insights from various environments within the human body. Stool samples remain the most common method, providing a rich source of microbial DNA that reflects the complex ecosystem residing in the intestines. This approach captures a multitude of organisms, revealing the diversity and abundance that characterizes gut health. Skin swabs represent another avenue for exploration, shedding light on the microbial communities inhabiting the surface of the body. This method uncovers the unique signatures of bacteria, fungi, and viruses, contributing to an understanding of skin health and conditions such as eczema or acne. By comparing samples from different body sites, researchers can elucidate how microbial populations vary, adapting to specific environments and influences. Saliva and oral rinses have also gained attention as less invasive options for sampling. These methods provide insights into the oral microbiome linked to both dental health and systemic diseases. Each sampling technique offers a distinct perspective, contributing to the broader narrative of microbial interactions and their implications for health. Emerging technologies, such as metagenomic sequencing, enhance the capacity to analyze these samples, revealing not only the identities of microorganisms, but also their functional potential. This knowledge marks a significant progression in understanding how these communities influence human biology. Metagenomics enables a deeper exploration of microbial DNA, revealing the vast genetic diversity harbored within these communities. By sequencing the genetic material extracted from environmental samples, researchers can identify and characterize microorganisms that evade traditional cultivation methods. This technique uncovers the hidden occupants of various ecosystems, from the human gut to extreme environments like hydrothermal vents. The applications of metagenomics extend far beyond mere identification. It offers insights into the functional capabilities of microbial communities, illuminating their roles in metabolic processes, nutrient cycling, and even disease modulation. For instance, Specific genes associated with antibiotic resistance can be traced, providing crucial information regarding the spread of resistance among microbial populations. Such analyses highlight the intricate interactions between microorganisms and their hosts, indicating how these entities can influence immune responses and overall health. The findings point to the microbiome's active participation in digestion and the synthesis of essential vitamins, showcasing its significance in maintaining homeostasis. In conjunction with advancements in bioinformatics, metagenomics fosters a comprehensive understanding of microbial ecology. Patterns of diversity, abundance, and genetic potential emerge, allowing researchers to visualize how these communities adapt to environmental changes and their implications for human health. This ongoing research continues to reshape our knowledge of the microbial world, underscoring its complexity and importance. Bioinformatics plays a critical role in microbiome research, facilitating the analysis and interpretation of vast quantities of data generated by advanced sequencing technologies. These tools enable scientists to decipher the intricate genetic blueprints of microbial communities, revealing their composition and functional potential. Each sample, collected from diverse environments, offers a unique snapshot of microbial life, showcasing the remarkable diversity that thrives within. Through computational methods, researchers can identify specific taxa and their relative abundances, constructing detailed profiles of microbial populations. Advanced algorithms then allow for the examination of relationships among these organisms, providing insights into their interactions and contributions to overall ecosystem dynamics. This meticulous analysis helps elucidate the roles these microbes play in human health, agriculture, and environmental sustainability. Data integration is also vital, as it combines findings from various studies and disciplines, fostering a more holistic understanding of microbiomes. By employing machine learning techniques, researchers enhance predictive modeling, leading to anticipatory insights regarding microbial behavior under different conditions. These sophisticated approaches enable the identification of potential biomarkers for diseases, dietary impacts, and environmental changes. Culturomics expands the boundaries of microbiome research by focusing on the vast majority of microorganisms that remain uncultured. Traditional methods often rely on cultivating organisms in laboratory settings, leaving a significant portion of microbial diversity unexamined. This innovative approach employs high-throughput sequencing and advanced bioinformatics to analyze genetic material directly from environmental samples. It allows for the detection of microorganisms that are otherwise challenging to isolate and grow. The findings from culturomics reveal a complex web of interactions within various ecosystems, including human hosts. Each microbiome is unique, shaped by individual genetics, lifestyle factors, and environmental conditions. This diversity contributes to the overall functionality of the microbiome, influencing health outcomes and responses to treatment. Analyzing uncultured microorganisms provides insights into their potential roles in processes such as digestion, immune regulation, and even mental health. The ability to characterize these hidden entities enhances understanding of microbial dynamics, offering a more comprehensive view of the living systems they inhabit. In clinical settings, this knowledge could lead to breakthroughs in personalized medicine, moving beyond generalized treatments. By considering the vast array of uncultured microorganisms, researchers can develop targeted interventions, optimizing health strategies for diverse populations. This unfolding field promises to reshape perceptions of microbial life and its impact on human existence. For synthetic biology represents a transformative approach to understanding and manipulating ecological systems, particularly within the microbiome. This discipline seeks to engineer microbial communities by designing organisms with specific functions, enhancing their roles in health and disease management. This region of research holds the potential to create tailored solutions for various health issues, such as obesity, diabetes, and even mental health disorders. Recreating custom microbial strains involves sophisticated techniques such as gene editing and metabolic engineering. These methods allow scientists to modify existing microbes or introduce new genetic material to enhance beneficial traits. The goal is to develop probiotics with increased efficacy, or to design microbes capable of producing therapeutic compounds directly within the human body. Experimental ecosystems within the microbiome can be monitored to study interactions and dependencies among species. This insight informs the design of robust microbial communities that may be more resilient to disturbances. By controlling these interactions, scientists can better predict outcomes in human health, contributing to personalized medicine. Such engineered microbiomes may also play a role in environmental sustainability. They can assist in bioremediation efforts by breaking down pollutants or enhancing soil health. This dual focus on human and environmental health underscores the expansive potential of synthetic biology to influence life on a microscopic scale, while fostering greater ecological balance. Fecal microbiota transplantation represents a promising frontier in the evolving landscape of microbiome research. This innovative procedure involves the transfer of fecal matter from a healthy donor to the gastrointestinal tract of a recipient. Through this process, a diverse array of beneficial microbes is introduced, aimed at restoring a balanced microbiome. Evidence suggests that such interventions can effectively treat conditions like recurrent Clostridioides difficile infections, demonstrating the powerful impact of microbial communities on human health. Research into FMT continues to expand beyond infectious diseases. Studies are investigating its potential role in addressing inflammatory bowel diseases, metabolic disorders, and even certain neurological conditions. The intricate relationship between the gut microbiome and the immune system hints at a broader applicability for this intervention. As understanding deepens, tailored approaches may emerge, where specific microbial profiles are selected to target individual health issues. Unethical considerations accompany these advancements. The sourcing of fecal material must adhere to rigorous screening processes to ensure safety and efficacy. Standardization of procedures is critical, as variability in donor microbiota can yield differing outcomes. Regulatory frameworks are evolving to keep pace with these developments, ensuring that FMT remains a reliable therapeutic option. Continued exploration of this field may unlock new therapeutic avenues, highlighting the profound influence of microbiomes on overall health and well-being. Personalized medicine represents a significant shift in healthcare, where treatments are customized based on individual microbiome profiles. This approach recognizes that the unique composition of microorganisms within each person can influence therapeutic responses. By analyzing these microbial communities, healthcare professionals can gain insights into how various treatments may be metabolized, thus optimizing efficacy and minimizing adverse effects. This precision allows for a more nuanced understanding of conditions such as obesity, diabetes, and autoimmune diseases. It is increasingly evident that the interplay between host and microbiome contributes to disease susceptibility and progression. Specific microbial signatures may serve as biomarkers, guiding clinicians in selecting the most appropriate interventions. Ongoing research is uncovering how dietary modifications and probiotics can augment therapeutic outcomes by reshaping the microbiome landscape. These interventions can enhance the body's natural resilience and restore balance, especially in individuals with dysbiosis. The prospect of utilizing microbiome analysis to inform treatment plans heralds a new era in the management of chronic diseases. This region of study continues to expand, potentially offering new insights into mental health disorders through the gut-brain axis. Understanding the connections between microbial populations and psychological well-being could lead to innovative treatment modalities, further demonstrating the microbiome's critical role in personalized medicine. Ethical considerations in microbiome research emerge as an essential area of focus, particularly given the profound implications for human health and societal norms. The collection and manipulation of microbial samples raise questions about consent, ownership, and privacy. Participants in studies may not fully grasp the complexities of their microbiomes, which can change over time and may have unforeseen consequences when altered. The potential for discrimination based on microbial profiles poses another ethical challenge. If specific microbiomes are linked to certain health conditions, it could lead to stigmatization or bias in healthcare settings. This region of inquiry emphasizes the need for robust frameworks that protect individual rights while fostering scientific advancement. Informed consent becomes paramount, as individuals must understand the risks and benefits associated with their participation in microbiome research. The implications of sharing microbial data extend beyond the individual, affecting communities and populations, necessitating a careful examination of collective privacy rights. Equitable access to microbiome-based therapies must also be considered, ensuring that advancements do not widen existing health disparities. Requires comprehensive policy development. This landscape of ethical inquiry serves to guide researchers and practitioners, encouraging a responsible approach as the field progresses toward unlocking the complexities of human microbiomes. Balancing innovation with ethical integrity remains crucial in shaping the future of personalized medicine. The intricate relationship between microbes and their human hosts has evolved over millennia, shaping not only individual health but also influencing the trajectory of human evolution. These microorganisms reside within various niches throughout the body, primarily in the gut, where they engage in a complex interplay with the immune system. This symbiotic relationship has fostered resilience against pathogens, enabling hosts to thrive in diverse environments. The colonization of these microbial communities begins at birth, with an initial influx of bacteria from the mother. Over time, this population diversifies, influenced by factors such as diet, environment, and lifestyle. Each individual harbors a unique microbiome, a reflection of their personal history and interactions with the external world. This uniqueness contributes to variations in metabolism, immune response, and even neurological functions. The co-evolution of humans and their microbiomes has also had profound implications for health and disease. Certain microbes may protect against chronic conditions, while imbalances can lead to dysbiosis, resulting in a spectrum of health issues. Understanding the genetic and functional capacity of these microbial inhabitants opens pathways to novel therapeutic approaches. By appreciating the nuanced roles they play, science continues to unravel the complexities of this hidden ecosystem, revealing the deep connections that define human existence. Microbial interactions within this ecosystem often manifest as mutualism and competition, two fundamental dynamics that shape the overall health and functionality of the microbiome. Mutualism occurs when different microbial species cooperate, providing benefits to one another. This relationship can enhance nutrient absorption, aid in digestion, and even stimulate the immune system. Such cooperation extends beyond individual species, creating intricate networks where each organism supports the other in a delicate balance. Competition, on the other hand, introduces a different dynamic. Microbes vie for limited resources, such as nutrients and space. This struggle can lead to shifts in community structure, influencing the overall composition of the microbiome. Some species may dominate, while others diminish. Understanding these relationships is crucial, as they illustrate how the microbiome adapts to changes within its environment. Factors such as diet, lifestyle, and external stresses can disrupt this equilibrium, leading to imbalances that may result in health issues. In the broader context of human health, the dual nature of microbial symbiosis highlights the importance of maintaining a diverse and stable microbiome. This stability serves as a foundation for resilience, supporting the complex interplay that sustains life at a microscopic level. Viruses play an intricate role in shaping the microbiome, often overlooked in discussions surrounding microbial life. They exist in vast numbers, outnumbering bacteria by a significant margin, influencing microbial dynamics within various environments. This prevalence allows them to drive evolutionary processes, altering bacterial populations through mechanisms such as horizontal gene transfer. Genetic material exchanged in this manner can confer advantageous traits, enhancing the survival of certain strains while diminishing others. This dynamic fosters a continually evolving landscape within the microbiome, where competition and cooperation coexist. Certain viruses specifically target bacteria, regulating their populations and preventing dominance by any single species. This regulation contributes to the overall balance and diversity that is essential for a robust microbiome. In addition to their regulatory functions, viruses can also enhance metabolic capabilities within microbial communities. By introducing new genetic material, they enable bacteria to access novel nutrient sources or adapt to changing environmental conditions. This adaptability is vital for maintaining equilibrium in ecosystems, including those within the human body. The interplay between viruses and the microbiome exemplifies a complex relationship characterized by constant change and adaptation. Understanding this relationship deepens insights into how microorganisms contribute to health and disease, revealing layers of interaction that continue to shape life at the smallest scales. Soil health relies significantly on the diverse communities of microorganisms that inhabit its depths. These microbes play a crucial role in nutrient cycling, decomposition, and organic matter formation, essential processes that sustain plant life and are consequently entire ecosystems. They enhance soil structure, improving aeration and water retention, vital for agricultural productivity. This region, rich in microbial inhabitants, acts as a dynamic reservoir of life, continuously interacting with both biotic and abiotic components. The balance of microbial populations can indicate soil vitality, with various species performing specialized functions. Nitrogen-fixing bacteria convert atmospheric nitrogen into forms accessible to plants, while fungi decompose complex organic materials, releasing essential nutrients back into the soil. The interplay among these microorganisms fosters a resilient environment capable of withstanding climatic fluctuations. Soil contamination or degradation can disrupt these delicate communities, leading to diminished fertility and increased vulnerability to pests and diseases. Sustainable agricultural practices, such as reduced tillage, crop rotation, and organic amendments, aim to preserve these microbial populations, ensuring that soil remains a thriving habitat. As knowledge of these microbial interactions expands, strategies for enhancing soil health emerge, promoting environmental sustainability. This quiet world, often overlooked, holds profound implications for agriculture and the broader ecosystem, emphasizing the interconnectedness of life. Microbial solutions for pollution represent a vital avenue for addressing environmental challenges. These microorganisms possess the remarkable ability to degrade or transform harmful pollutants, making them invaluable allies in the quest for cleaner ecosystems. In contaminated environments, these agents initiate processes that break down complex toxic compounds into simpler, less harmful substances. Certain species thrive in extreme conditions, such as heavy metal-laden soils or oil spills, where their resilience enables them to metabolize contaminants that would be detrimental to most forms of life. This capacity for bioremediation serves not only to restore the health of affected areas, but also to enhance soil fertility and promote biodiversity. Case studies reveal the efficacy of these organisms in various applications, from wastewater treatment facilities to oil recovery operations. In these scenarios, microbial communities work collaboratively, often forming intricate networks that optimize pollutant degradation. The implications extend beyond immediate remediation efforts. Understanding these processes can inform strategies for sustainable development. By harnessing the power of microorganisms, industries can minimize their ecological footprint, paving the way for innovative solutions that align with natural cycles. As research advances, the potential for engineered microbes to target specific pollutants becomes increasingly feasible, promising a future where pollution is managed with precision and efficiency. This approach highlights the significance of microbiomes in shaping a healthier planet. Microbiomes thrive in extreme environments, offering insights into resilience and adaptability in the harshest conditions, such as hydrothermal vents and polar ice caps. These microorganisms exhibit remarkable survival strategies. High temperatures, intense pressures, and salinity create an environment where only the most specialized organisms can flourish. At hydrothermal vents, chemosynthetic bacteria harness energy from inorganic compounds, converting it into organic matter. This process forms the foundation of entire ecosystems, supporting a variety of life forms that would otherwise not exist in such desolate areas. These microorganisms demonstrate a unique ability to withstand extreme temperatures, with some species thriving at temperatures exceeding 300 degrees Celsius. In the icy realms of Antarctica, psychrophilic bacteria have adapted to survive in temperatures well below freezing. These organisms produce antifreeze proteins, allowing them to maintain cellular functions in sub-zero conditions. The study of these microbiomes reveals not only their resilience, but also the potential applications in biotechnology and medicine. By understanding how these organisms thrive, researchers can unlock new avenues for innovation, from developing sustainable energy sources to creating more effective pharmaceuticals. The examination of these unique communities illustrates the profound adaptability of life, serving as a testament to nature's ingenuity in overcoming the most formidable challenges. The lessons learned from these extreme microbiomes are invaluable in the quest to understand life itself. Variations in the microbiome of animals reveal fascinating parallels and distinctions with the human microbiome. Each species maintains a unique microbial community, shaped by diet, habitat, and evolutionary history. This region of complex interactions offers insight into how these microorganisms influence health and disease across diverse organisms. In herbivores, for instance, specialized microbes assist in breaking down cellulose, allowing access to nutrients that would otherwise remain locked within plant material. Their adaptations showcase an intricate balance between host and microbiota, often resulting in mutualistic relationships that enhance survival. Similar mechanisms exist within the human gut, where a diverse array of bacteria aids in digestion and nutrient absorption. Carnivorous animals present another intriguing picture. Their microbiomes often reflect the high protein content of their diets, with specific bacteria adapted to metabolize meat. This adaptation highlights the role of microbial communities in facilitating nutritional needs, echoing the functionality seen in human microbiomes influenced by dietary patterns. The relationship between these microbiomes extends beyond mere digestion. They partake in immune system modulation, protecting hosts from pathogens. This interplay emphasizes the importance of microbial diversity and its implications for understanding health across species. The intricate connections within these ecosystems extend to the realm of agriculture, where the microbiome plays a pivotal role in plant health. These communities of microorganisms inhabit the soil, forming complex relationships with plant roots. They assist in nutrient absorption, facilitating access to essential elements such as nitrogen, phosphorus, and potassium. This symbiosis enhances plant resilience, enabling crops to withstand environmental stresses. In this region, beneficial bacteria and fungi create a protective barrier around roots, preventing pathogenic organisms from establishing themselves. This defense mechanism not only improves crop yield, but also reduces the reliance on chemical fertilizers and pesticides, promoting more sustainable agricultural practices. The diversity of microbial life contributes significantly to soil health, influencing its structure and fertility. Research has shown that specific microbial strains can enhance plant growth by producing plant hormones and metabolites. These interactions stimulate root development and improve overall plant vigor. The application of microbial inoculants has emerged as a promising strategy to boost agricultural productivity while minimizing environmental impact. Within controlled environments, such as greenhouses, the manipulation of microbiomes has demonstrated the potential to optimize plant growth conditions. Continuous exploration of these microbial networks may reveal further insights into enhancing food security and agricultural sustainability, highlighting their vital role in modern farming practices. Freshwater and marine microbiomes serve as foundational components of aquatic ecosystems, engaging in complex interactions that support various forms of life. These microorganisms thrive in diverse environments, ranging from tranquil lakes to the vastness of oceanic expanses. The unique conditions of each habitat shape the composition and behavior of these microbial communities, influencing nutrient cycling and energy flow. In freshwater systems, it is the balance between autotrophic and heterotrophic organisms that maintains ecological stability. Phytoplankton, for instance, harness sunlight to produce energy, while bacteria decompose organic matter, recycling nutrients back into the ecosystem. This interplay fosters a dynamic environment where productivity is closely tied to microbial activity. Marine microbiomes, characterized by their vastness and complexity, contribute significantly to global biogeochemical cycles. They play a pivotal role in carbon fixation, contributing to the ocean's ability to sequester carbon dioxide. The interactions between bacteria, archaea, and viruses in these waters create intricate networks that affect not only marine organisms, but also the climate as a whole. Both freshwater and marine environments exhibit a remarkable degree of resilience and adaptability, often responding to environmental changes with shifts in microbial community structure. Understanding these intricate dynamics is essential for managing aquatic resources and addressing challenges posed by pollution and climate change. The impact of climate change on microbial communities is profound and multifaceted. Elevated temperatures alter metabolic rates, leading to shifts in community composition. In warmer environments, certain microorganisms may thrive, while others diminish, disrupting established ecological balances. These alterations can result in changes to nutrient cycling, affecting the availability of crucial elements like nitrogen and phosphorus within ecosystems. Increased carbon dioxide levels foster the growth of specific groups, particularly those adept at utilizing this abundant resource. This phenomenon can enhance primary productivity, but may simultaneously lead to an imbalance, as some species outcompete their counterparts. The interactions between microorganisms and their environments become increasingly complex under these shifting conditions. Oceanic systems experience rising temperatures, which can cause stratification. This process limits nutrient mixing, impacting the diversity and abundance of microbial life. In these regions, the alteration of food webs becomes evident, influencing higher trophic levels and the overall health of marine ecosystems. Freshwater habitats face similar threats. Changes in precipitation patterns can lead to altered flow regimes, affecting microbial communities in rivers and lakes. With these modifications, the resilience of these microbial populations is tested, posing a risk to ecosystem functions that rely on their stability. Overall, the delicate interplay between climate change and microbial dynamics underscores the urgent need for monitoring and understanding these vital organisms. Microgravity environments present unique conditions that alter microbial behavior, revealing insights that are often obscured on Earth. In the absence of gravitational forces, these organisms exhibit changes in growth patterns, gene expression, and interactions with one another. This region, where weightlessness reigns, becomes a laboratory for studying microbial resilience and adaptability. Experiments conducted aboard the International Space Station highlight how microbes can thrive in extreme environments, often exhibiting enhanced growth rates and altered virulence. These findings challenge preconceived notions of microbial limitations, suggesting that life can persist and evolve under conditions previously deemed uninhabitable. The effects of microgravity extend beyond mere growth. They also influence the microbial community structures. In this altered ecological setting, certain species may dominate, leading to shifts in functionality and metabolic pathways. The implications for human health are profound. Understanding these changes is crucial, particularly for astronauts who may face heightened susceptibility to infections during prolonged space missions. Research into the microbiome in space also informs astrobiology, shedding light on potential life forms beyond Earth. Insights gained from these studies contribute to our understanding of life's tenacity and adaptability, reinforcing the notion that microbial life is an integral part of the cosmic landscape. This exploration of the hidden world within and beyond enhances the narrative of life itself, forging connections across the vastness of space. Citizen science has emerged as a pivotal force in microbiome research, fostering collaboration between professional scientists and enthusiastic individuals. This collective effort expands the pool of data, allowing for a broader understanding of microbial diversity and function. Engaged citizens contribute to research projects by collecting samples from their environments, whether it be soil, water, or even their own bodies. Each sample adds to the growing repository of knowledge, revealing intricate patterns and relationships within microbial communities. The process of data collection often involves simple methodologies, making it accessible to a wide audience. Participants may use standardized kits to analyze their samples, which are subsequently shared with researchers. This interaction creates a dynamic feedback loop, where findings from citizen science projects can inform professional studies, enhancing the overall understanding of microbial ecosystems. Communities benefit from this engagement, as individuals learn about the significance of their local environments and the microorganisms that inhabit them. Through educational programs and workshops, awareness of microbiomes and their ecological roles grows. This grassroots movement not only democratizes scientific research, but also inspires a sense of stewardship for the environment. As knowledge proliferates, the intricate connections between human health, ecological balance, and microbial life become increasingly evident, underscoring the importance of collaboration in scientific discovery. The implications for public health policy emerge as a crucial consideration in light of this knowledge. Microbial communities within the human body influence everything from digestion to immunity, shaping individual health outcomes. Policies that prioritize microbiome research can lead to enhanced understanding of disease prevention and management. This region of study underscores the potential for innovative strategies in addressing chronic conditions and infectious diseases. Investment in microbiome research may yield significant benefits for public health systems, guiding the development of probiotics, prebiotics, and other therapeutic interventions. These approaches can promote gut health, enhance immune responses, and reduce healthcare costs associated with chronic illnesses. Policy frameworks that support interdisciplinary collaboration can facilitate the integration of microbiome science into clinical practices. Education plays a vital role in public health initiatives. Informing communities about the microbiome's role fosters awareness of lifestyle choices that impact microbial diversity. Encouraging a balanced diet rich in fruits, vegetables, and fermented foods may support healthier microbiomes, contributing to overall well-being. As the understanding of microbiomes evolves, so too must the policies that govern health systems. Regulatory frameworks should adapt to incorporate microbiome research findings, ensuring that emerging therapies are safe and effective.
This quiet revolution in science has far-reaching implications for individual and community health, necessitating a proactive approach to policy development. Educational outreach plays a crucial role in disseminating knowledge about the microbiome. It serves as a bridge between scientific research and public understanding, fostering a culture of awareness. Workshops and seminars aim to engage diverse audiences, from healthcare professionals to school children. These initiatives provide insights into the various roles that microbial communities play within the human body, highlighting their impact on digestion, immunity, and even mental health. Multimedia campaigns utilize platforms ranging from social media to documentaries, bringing attention to the unseen inhabitants that shape our well-being. Infographics and interactive tools simplify complex information, making it accessible to those without a scientific background. Schools incorporate microbiome education into curricula, encouraging students to explore the microscopic world through hands-on experiments and discussions. Community gardens and local health initiatives promote the importance of nutrition, emphasizing the connection between diet and microbiome health. This approach nurtures a holistic understanding of wellness, where individuals recognize the significance of their dietary choices on their internal ecosystems. Collaborations between scientists and community leaders further expand outreach efforts, ensuring that information reaches underserved populations. By cultivating this awareness, individuals become empowered to make informed decisions about their health. The collective effort to understand and respect the microbiome will shape future generations, creating healthier communities built on the foundation of knowledge. This intersection between technology and microbiome studies has opened new frontiers in scientific inquiry. Advanced sequencing techniques, such as metagenomics, allow researchers to identify and catalog the vast array of microbial species inhabiting diverse environments. These methods generate immense quantities of data, revealing intricate patterns of microbial interactions and their influence on human health and the ecosystem. Innovative bioinformatics tools play a crucial role in parsing this data, transforming complex biological information into accessible frameworks. Machine learning algorithms analyze microbial communities, predicting how shifts in composition might correlate with health outcomes or environmental changes. These technologies not only enhance understanding, but also facilitate the development of personalized medicine, tailoring interventions based on an individual's unique microbiome profile. In clinical settings, real-time monitoring devices measure microbial metabolites in bodily fluids, providing immediate insights into metabolic processes influenced by the microbiome. Such advancements support the design of targeted therapies, which can mitigate diseases linked to microbial imbalances. These insights extend beyond human health, impacting agriculture and environmental management. By harnessing microbial knowledge, sustainable practices can be developed, enhancing soil health and crop resilience. The synthesis of technology and microbiome studies fosters a deeper comprehension of life's interconnectedness, paving the way for innovative solutions to complex challenges. Historical practices in traditional medicine often recognized the importance of microbial life, long before modern science provided clarity. Ancient cultures utilized various herbal remedies, fermented foods, and rituals aimed at nurturing the body's internal ecosystem. These methods reflect an intrinsic understanding of balance and harmony within the human body. In many indigenous practices, the concept of wellness relied heavily on the cultivation of beneficial microorganisms. Fermented beverages, such as kefir and kombucha, were not only valued for their flavors but also for their ability to promote gut health. This region of the body, often referred to as the second brain, is a vital component of overall health. It houses trillions of microorganisms that influence digestion, immunity, and even mood. Traditional healers recognized that disturbances in this delicate balance could lead to illness. Various remedies aimed at restoring equilibrium included herbal concoctions and dietary adjustments, often tailored to individual needs. These approaches mirrored the scientific understanding of microbial diversity and its role in maintaining health. Such practices highlight a long-standing relationship between humans and their microbial partners. The insights gained from studying these historical methods offer valuable perspectives on contemporary healthcare. They underscore the potential for integrating ancient wisdom with modern science to foster holistic health solutions. Emerging technologies are poised to illuminate the intricate dynamics of the microbiome. Advancements in sequencing techniques, particularly metagenomics, allow for unprecedented insights into the vast array of microbial species residing within various environments, including the human body. This capability facilitates the identification and characterization of previously unknown organisms, advancing the understanding of their roles in health and disease. The application of artificial intelligence is transforming data analysis in microbiome research. Through machine learning algorithms, researchers can now analyze complex datasets at remarkable speed and accuracy. This technology aids in predicting interactions between microbial communities and their hosts, potentially leading to personalized therapeutic strategies. Another promising avenue involves the development of targeted probiotics and prebiotics. By harnessing genomic and metabolomic data, scientists can design specific microbial formulations tailored to individual needs, optimizing gut health and overall well-being. This precision approach may mitigate the effects of dysbiosis and contribute to the prevention of various diseases. Environmental microbiome studies are also expanding, revealing the interconnectedness of ecosystems. By understanding how microbial communities respond to climate change and pollution, it becomes possible to devise strategies for ecological preservation. Continued exploration in these domains will deepen the comprehension of the microbiome's influence, paving the way for innovative solutions in both health and environmental sustainability. Educational initiatives around microbiomes are crucial for fostering microbial literacy among the general population. This understanding allows individuals to engage with scientific discussions surrounding health and environmental issues. When communities grasp the fundamental principles governing these microscopic ecosystems, they become empowered to advocate for informed decision-making in public policies and personal health practices. Increased awareness can bridge the gap between scientific research and everyday life. Individuals equipped with knowledge about the role of microbes in digestion, immunity, and even mood regulation can make choices that enhance their well-being. Such literacy promotes a deeper appreciation of the interconnectedness of human and environmental health, recognizing that the vitality of one often hinges on the other. Engagement in discussions about antibiotic use, for instance, can lead to more responsible practices that preserve microbial diversity. This diversity is vital for resilience against diseases and environmental changes. Civic discourse that incorporates microbial literacy encourages a collaborative approach to public health, where citizens and scientists work together to confront challenges like antibiotic resistance and climate change. By fostering a culture of understanding, communities can cultivate a more sustainable future, ensuring that the insights gained from microbiome research translate into tangible benefits for society at large. Such an approach promises to reshape health narratives and environmental stewardship. The intricate interplay between the microbiome and mental health reflects a burgeoning field known as nutritional psychiatry. This area examines how dietary choices influence emotional well-being, revealing that specific foods can foster a flourishing microbial community. The presence of diverse bacteria informs various biochemical pathways that affect neurotransmitter production, particularly serotonin, often referred to as the feel-good hormone. Research indicates that a balanced, nutrient-rich diet, rich in fiber, prebiotics, and probiotics, cultivates a diverse microbiota. This diverse population enhances resilience against stress and anxiety. Foods like fermented products, whole grains, and a variety of fruits and vegetables contribute to this microbial diversity, creating an environment conducive to emotional stability and cognitive function. Conversely, diets high in processed sugars and unhealthy fats may lead to dysbiosis, an imbalance that has been correlated with increased instances of mood disorders. The connection between the gut and brain, often termed the gut-brain axis, underscores the importance of maintaining a healthy microbiome to support mental health. Emerging studies highlight that dietary modifications can serve as a therapeutic approach, potentially reducing symptoms of depression and anxiety. The implications are profound, suggesting that personalized nutrition, tailored to enhance microbiome health, may play a critical role in holistic mental health strategies. Chronic illnesses often emerge from complex interactions within the body, and the microbiome plays a pivotal role in these dynamics. This collection of microorganisms, residing primarily in the gut, influences inflammation, metabolism, and immune responses. The intricate balance of beneficial and harmful microbes can significantly impact chronic conditions such as diabetes, cardiovascular diseases, and autoimmune disorders. Imbalances in this microbial community may lead to systemic inflammation, which is increasingly recognized as a contributor to various chronic ailments. Research indicates that specific strains of bacteria can either mitigate or exacerbate these inflammatory pathways. For instance, the The presence of certain beneficial microbes has been associated with improved metabolic health, while their absence correlates with increased risk of metabolic syndrome. Holistic approaches to chronic illness often involve interventions that promote a healthy microbiome. Nutritional strategies, including the incorporation of prebiotics and probiotics, can help restore microbial balance. Additionally, lifestyle factors such as stress management, sleep quality, and regular physical activity are essential in nurturing this region of biological activity. Thus, understanding the microbiome not only offers insights into the underlying mechanisms of chronic illness, but also provides avenues for prevention and management. This scientific exploration continues to unveil the silent yet impactful relationship between microbial health and overall well-being. Community microbiomes serve as intricate networks where diverse microbial populations interact in complex ways. These networks are not merely collections of individual species, they function collectively, influencing each other's behavior and health. Within these ecosystems, competition and cooperation coexist, shaping the dynamics of microbial life. Interactions between different microbial species can enhance nutrient availability, enabling them to thrive in environments where resources may be limited. This synergy plays a crucial role in nutrient cycling, contributing to the overall health of their host.
In human bodies, these communities contribute to metabolic processes, synthesizing essential vitamins and breaking down complex carbohydrates that the host cannot digest alone. In various environments, from soil to the human gut, microbial networks exhibit resilience. This resilience allows them to adapt to changing conditions, such as fluctuations in pH or temperature, which might adversely affect individual species. The stability of these networks can be essential for maintaining ecological balance, ensuring that beneficial microbes flourish while pathogenic organisms are kept in check. This intricate interplay of microbes is visible in various ecosystems, including those found in the human microbiome. Each community reflects a unique composition shaped by genetics, diet, and environment. Understanding these dynamics offers insights into how they contribute to health and disease, emphasizing the importance of microbial diversity and interaction. Strategies for a healthy future increasingly hinge on the intricate relationships within this microscopic community. The role of the microbiome in disease prevention is becoming clearer as research reveals potential pathways through which these organisms influence immune function and metabolic processes. A balanced microbiome can fortify the body against pathogens, reducing the likelihood of infections and chronic illnesses. Diet plays a pivotal role in shaping this microbial environment. Consumption of diverse, nutrient-rich foods fosters a robust population of beneficial bacteria. Fermented products, high-fiber offerings, and a variety of fruits and vegetables cultivate an ecosystem that thrives on diversity, creating a barrier against harmful microorganisms. Probiotics and prebiotics have emerged as promising interventions to enhance microbial health. Probiotics introduce beneficial strains, while prebiotics serve as nourishment for these microorganisms, allowing them to flourish. Research supports the idea that these measures can bolster gut health, improving resilience against diseases such as obesity, diabetes, and inflammatory conditions. The environment also influences microbiome composition. Exposure to diverse ecosystems, whether through nature or varied living conditions, has shown benefits in microbial richness. Understanding the interplay between lifestyle choices and the microbiome provides a foundation for strategies aimed at promoting long-term health and preventing disease, highlighting the intricate balance of life within and its profound implications for well-being. Personalized nutrition leverages insights from the microbiome to tailor dietary choices that enhance individual health. This approach recognizes that each person's unique microbial community influences how nutrients are processed and utilized. Distinct variations in microbial composition can affect metabolism, making certain foods more beneficial or detrimental to specific individuals. The relationship between diet and the microbiome is dynamic. Certain foods can nourish beneficial bacteria while inhibiting harmful strains. These interactions can lead to significant shifts in overall health, impacting everything from inflammation levels to mood stability. By analyzing an individual's microbiome, nutritionists can recommend specific dietary adjustments that promote a favorable environment for beneficial microorganisms. Emerging research highlights the importance of fiber-rich foods, fermented products, and polyphenol-containing items in cultivating a diverse microbiome. This diversity is crucial, as it contributes to resilience against diseases and enhances metabolic flexibility. Custom diets, informed by microbial analyses, can aid in weight management, improve digestive health, and even bolster immune function. The implications of such personalized strategies extend beyond mere dietary preferences. They offer a glimpse into a future where nutrition is not one size fits all, but rather a finely tuned orchestration of food choices based on individual microbial landscapes. This nuanced understanding underscores the potential for personalized nutrition to transform health outcomes on a broad scale. The vast integration of microbiome research into skincare products is gaining momentum. This burgeoning market reflects a growing recognition of the skin's microbiome as an essential component of overall skin health. Various products now emphasize the inclusion of prebiotics and probiotics, aiming to restore and maintain the delicate balance of microbial communities residing on the skin's surface. Brands are increasingly formulating their offerings with an emphasis on the skin's natural barrier. These formulations often include beneficial microbes that aim to enhance skin resilience and hydration. The idea is not merely to cleanse and beautify, but to support the skin's inherent microbiological ecosystem, fostering a healthier complexion over time. Consumer awareness is shifting towards products that provide holistic benefits rather than superficial fixes. This change is evident in the rising demand for products that promote microbiome health, as individuals seek solutions that address underlying issues such as irritation and sensitivity. Market trends indicate a robust growth trajectory for microbiome-focused skincare, driven by scientific advancements and consumer education. The evolving landscape reveals a shift from traditional formulations to those that recognize the skin as a living organ, influenced by its microbial inhabitants. The promise of restoring balance within this ecosystem presents a new frontier in skincare, blending nature with modern science. The intricate dynamics of microbial resistance reveal a complex interplay of organisms residing within. Antibiotic resistance occurs when these microorganisms evolve mechanisms to withstand the effects of antimicrobial agents. This phenomenon is not merely a consequence of medical intervention, but rather a natural evolutionary process. Within this realm, certain bacteria possess genetic adaptations that allow them to survive despite the presence of antibiotics, leading to the emergence of resistant strains. These strains can proliferate rapidly, particularly in environments where antibiotic usage is prevalent. The result is an alarming increase in infections that are difficult to treat, raising public health concerns globally. Resistance can spread through horizontal gene transfer, where genetic material is exchanged between different species, further complicating the landscape of microbial interactions. This region of the microbiome serves as a battleground where bacteria constantly adapt to their environment, including the selective pressures imposed by antimicrobial substances. Understanding these mechanisms provides valuable insights into the delicate balance of microbial communities and their resilience. Research continues to explore potential strategies for mitigating the impacts of resistance. Approaches such as phage therapy and the development of new antimicrobials aim to counteract resistant organisms while preserving beneficial bacteria. The future of healthcare hinges on recognizing the significance of these microbial inhabitants and their roles in our overall well-being. Innovations in biotechnology are increasingly informed by the intricate workings of the microbiome. Research highlights the potential of these microorganisms to revolutionize various fields, from medicine to environmental science. By harnessing their capabilities, scientists are developing new therapeutic strategies that target specific health issues, leveraging the natural processes these microbes employ. Synthetic biology is one area witnessing remarkable advancements. Through genetic engineering, it becomes possible to design microbial strains that can produce pharmaceuticals more efficiently. This approach not only reduces manufacturing costs, but also minimizes the environmental impact associated with traditional production methods. The ability of certain microbes to metabolize complex compounds opens pathways for bioremediation, effectively addressing pollution in diverse ecosystems. In agriculture, the understanding of soil microbiomes leads to innovative practices that enhance crop resilience by promoting beneficial microbes within the soil. Farmers can improve nutrient uptake and reduce reliance on synthetic fertilizers. This transformation fosters sustainable practices, contributing to global food security. The exploration of the gut-brain axis reveals further opportunities for innovation. Insights into how these microorganisms influence mental health encourage the development of probiotic therapies. Such advancements suggest a deeper connection between physical health and microbial diversity, paving the way for holistic approaches to wellness. And this evolving landscape, the microbiome stands as a beacon of potential, reshaping the boundaries of biotechnology and health. Technical hurdles present significant challenges in microbiome research, often stemming from the complexity and variability of microbial communities. These communities exhibit immense diversity, with trillions of microorganisms existing within a single individual. Analyzing this myriad of species requires advanced sequencing technologies and computational tools capable of handling vast datasets. The need for standardized methodologies complicates comparisons across studies, hindering the ability to draw definitive conclusions. Issues related to sample collection, processing, and storage further complicate the reliability of findings. Ethical considerations also loom large in this emerging field. Manipulating microbial populations within the human body raises fundamental questions regarding consent and the implications of such interventions. The potential for unintended consequences emphasizes the importance of rigorous ethical oversight. Balancing innovation with responsible research practices becomes essential as scientists navigate these uncharted territories. In clinical applications, the use of probiotics or fecal transplants carries inherent risks, necessitating thorough evaluation of safety and efficacy. Regulatory frameworks must evolve to address the unique challenges posed by microbiome-related therapies, ensuring that advancements do not outpace ethical considerations. The intersection of technology and ethics propels the field forward, revealing both the promise of microbial intervention and the caution required in its pursuit. Each step taken must be grounded in a commitment to understanding these intricate ecosystems and their profound impact on human health. Research into microbiomes has unveiled a myriad of connections between these microscopic communities and global health trends. Scientists are increasingly recognizing the role of these organisms in numerous conditions, including obesity, diabetes, and mental health disorders. This understanding has fostered international collaboration, leading to expansive databases that catalog microbial species and their functions, Such efforts are pivotal in identifying patterns that transcend geographical boundaries. In clinical settings, advancements in microbiome therapies are emerging. Probiotics and personalized microbiome interventions are being tailored to individual patients, aiming to restore balance within these communities. This approach highlights the importance of recognizing variability among populations, which can influence treatment efficacy. The environmental impact of microbiomes is also gaining attention. Soil and aquatic microbiomes play crucial roles in nutrient cycling and ecosystem health. Research focused on these domains promises insights into sustainable practices that could mitigate climate change effects. Future directions in microbiome studies are likely to emphasize interdisciplinary approaches, merging insights from genomics, metabolomics, and ecology. Such synergies could uncover novel interactions between microbiomes and their environments, providing a deeper understanding of their influence on global health and climate resilience. As this field advances, it holds the potential to reshape public health policies and environmental strategies worldwide. This new frontier in science reveals an intricate world of microorganisms that inhabit various ecosystems, including the human body. These communities, composed of bacteria, viruses, fungi, and archaea, play crucial roles in maintaining health and homeostasis. The sheer diversity of these organisms reflects a complex interplay of genetic, environmental, and dietary factors, shaping individual microbiomes uniquely. Research indicates that these microbial populations influence metabolism, immune responses, and even mental health. The composition of this microbiome can be altered through external factors such as nutrition, medication, and lifestyle choices. This adaptability opens avenues for therapeutic interventions, such as microbiome transplantation, which aims to restore balance in cases of dysbiosis, a condition associated with various diseases. Current studies explore the microbiome's interaction with the environment, revealing how external pollutants may impact microbial health and in turn human health. This relationship underscores the interconnectedness of ecosystems, as the health of one system may directly affect another. Technological advancements, including metagenomic sequencing, allow for the in-depth analysis of these communities, uncovering their functions and potential benefits. As understanding deepens, it becomes clear that the microbiome is not merely a collection of microorganisms, but a vital component of biological systems, deserving careful consideration in scientific inquiry and health-related policies. This intricate community of microorganisms plays a crucial role in maintaining homeostasis within the human body. Each interaction between these entities and human cells contributes to a delicate balance that influences numerous physiological processes. It aids in digestion, synthesizes essential vitamins, and strengthens the immune system, showcasing its multifaceted contributions to overall health. This imbalance in the ecosystem can lead to significant consequences. Dysbiosis, or an imbalance of microbial populations, has been implicated in various conditions, including obesity, diabetes, and inflammatory bowel disease. These associations highlight the potential for microbiome analysis to serve as a biomarker for disease susceptibility, paving the way for personalized medicine approaches that target specific microbial profiles. Research continues to unveil the complex interactions between the microbiome and other biological systems, such as the nervous system. The gut-brain axis illustrates how microbial metabolites can influence mood and behavior, suggesting that psychological health may also be intertwined with these microscopic inhabitants. As scientists delve deeper into this hidden world, the implications for therapeutic interventions grow. Probiotics, prebiotics, and dietary modifications are being explored as strategies to restore balance and promote health. This evolving understanding emphasizes the importance of nurturing the microbiome, recognizing it as an essential ally in the pursuit of holistic health.